Hi there everyone. Today we're going to be talking about tissue development. As always, please make sure you've read the essential reading that's been recommended for this section on Blackboard. There's a section of the Ross and Wilson textbook to have a look at, and also a PDF on tissue types that I asked you to look at. So hopefully as we go through this presentation, that'll all start to make a bit more sense to you. So the aim of this is to explain the origin of tissues. Now that sounds rather dramatic. Don't worry, we're not going to be getting into evolution versus intelligent design or anything like that. What we're looking at is where human tissue develops from an embryological point of view, so from conception onwards. This is a huge topic, embryology. You know, there's people that are quite simply embryologists. We're looking at this at a very basic level. And it's just so you understand roughly, very basically, where the different tissues that we're going to be talking about over the next few months in this module have originated from. And we're also going to have a little look at tissue types, that being epithelium connective tissue and muscle and nerve tissue. You'll probably have noticed from the lecture list that we go on to talk about these in much more detail. So again, it's just sort of a brief overview of that today. So starting then with embryonic development, there's an awful lot goes on, as you might expect, in embryonic development, and a huge amount of that goes on within the first trimester. So everything that I've got in that stages diagram, list even here, all this here, first trimester. So the first 13 weeks. It's a really, really important stage of pregnancy. And interestingly enough, or something that I find interesting, or maybe even a bit strange to think about, is that the vast majority of this actually goes on before a woman even knows she's pregnant. You'll notice we've got 1, 2, 3, 4 things up to Blastocyst there before we're even at 7 days. So a huge amount goes on early on. So we're going to talk about that first, and then we can have a look at sort of second and third trimester. So very early on, obviously right at the beginning, we've got fertilisation. So you've got fusing of the male and female sex cells. So ovum being the female sex cell, and spermatozoon would be the proper full word for sperm or the male sex cell. We tend to refer to them as gametes at this stage. So an unfertilised sex cell is a gamete. So once they fuse, that's when you get fertilisation. And from that stage, we refer to the fertilised ovum as a zygote. So you can see from this diagram here that fertilisation at the very first stage is actually happening quite far up. And people often think, obviously, sorry, I should probably clarify first, we're looking here at a diagram of a womb with an ovary and a fallopian tube connected them. I'm sure you're all aware of what that diagram is already, but just to clarify. So often people think that fertilisation goes on in the womb, but actually it's way up just as the egg is released from the ovary in the fallopian tube that this happens. So from then on, we refer to that as zygote. Obviously in humans and in most mammals, well, all bar one, fertilisation happens internally. Other animal species, like fish, for example, you've got external fertilisation, so the female fish would lay her eggs and then a male fish would come along and spread his sperm over the top of them, so it happens externally. But in humans, we're talking internally. And it can be engineered through IVF. So during IVF, in general, the fertilisation process and the formation of a zygote occurs outside the body, and then it's implanted into the womb. So at that point, you have just one cell, because if you remember when we talked about cell division, as opposed to mitosis, where you have two identical cells separate, when sex cells are created by meiosis, you get two cells with half the amount of genetic material. So that means that these gametes, the ovum and the sperm, have only got half the amount of genetic material, and therefore they combine to form one cell. It's not two cells at that point, it's one cell. So we need to obviously develop this into multiple cells in order to form the baby that's developing. So as the zygote travels along the fallopian tube towards the womb, a process of cleavage happens. So cleavage is when the zygote rapidly divides to produce multiple cells, and you can see that from this image here. It's divided to 1, to 4, to 8, and so on. It's mitotic division, so each cell is dividing to form 2 identical cells, and it happens about every 20 hours. So every 20 hours you've got double the amount of cells that you had previously, so hence 2, 4, 8, 16, and so on and so on. The cells themselves are referred to as blastomeres. And at this point, the zygote never gets any bigger. So it started off as a really big cell. An ovum is a huge, big cell. And it's divided within there. So multiple cells, but they've got smaller and smaller and smaller. And when we get to the point where we have at least 16 cells, so after the 4th division, we then have a morula. That's what we've got here. And it is a morula when it exits the fallopian tube and enters the uterine cavity, so enters into the womb. This has all happened really quickly. At around 4 days after fertilization, so very few people would know they were pregnant at that stage. Just 4 days after fertilization, the morula is into the uterine cavity, and it develops a little hole, like a little cavity within the morula, and that goes on to develop other things which we're going to talk about. So just to write down a couple of terms there that I've introduced that are not written down. Excuse me whilst I add a slide in, and we can add there. We talked about blastomere. So that was the cells that are formed within the zygote as a process of cleavage. In fact, we talked about cleavage as well, so let's put that there. So cleavage is the process by which the cells divide. Blastomere is the cells that are produced as a result of cleavage. I won't write morula because that's on the image on the slide before. But we did talk about the cavity that develops within the morula at about 4 days, which is known as a blastocyst. So, continuing on, we've now got the morula into the uterine cavity, and it's developed the blastocyst. It continues to divide and divide, and after the 7th cleavage, which means there's 128 cells, it becomes a blastocyst. Again, I'll write that down because it's not written in the images. Blastocyst. So blastocyte was the cavity within the morula. And now that we've got 128 cells, it stops being a morula and becomes a blastocyst. And 2 layers form in the blastocyst, and that's what this image is showing here. Okay, this is the blastocyst. In fact, let's put an arrow there so that's clear. That's not an arrow, it's a line, but I'm sure you understand what I mean. So the blastocyst develops into 2 layers. We have an outer layer of cells and an inner layer of cells. So the outer layer of cells is known as the trophoblast, and it's in contact with the uterine wall. And the inner layer of cells, which is shown in this little image here, is known as the embryoblast. So it's the trophoblast on the outside of the blastocyst that assists in the implantation because it's in contact with the uterine wall. And the trophoblast actually creates enzymes which break down the lining, the uterine lining, and allow the cells of the blastocyst to become embedded. And very quickly you start to get capillaries develop between the blastocyst and the uterine wall. So the blastocyst becomes part of the uterine wall. And up until this point, it's just been floating around, but now it's implanted. This is the stage at which pregnancy hormones rise, because up until this point, the body hasn't necessarily known it's pregnant because it's just been floating around. But now that there's implantation, you get pregnancy hormones rise, the body's aware, and this is the point at which a pregnancy test will generally give or begin to give a positive result. So we're still looking at around 4 to 6 days. We're not quite 7 yet. So this image here is showing us a little bit further on in the implantation process. You can see that the uterine wall is around the outside here, and we have the blastocyst that's well embedded into the uterine wall. So implantation is complete. We've got circulation between the uterine lining and the blastocyst, and it starts to develop into a disc with 2 layers to the disc. So you can see from this image here, we've got a round disc, but we've got sort of a large part here and then a smaller part here. We have what will now develop into the embryo, so that's the larger part, and then we have the amniotic cavity, which can also be known as the yolk sac. So embryo being the biggest part and the yolk sac, or the amniotic cavity. And even this early on, we're starting to get an organisation of circulation. It's not just random capillary beds. They're starting to organise themselves into a particular arrangement for future development. Following on from here, around about 16 days, so just over 2 weeks from fertilisation, we've got a process of gastrulation. Gastrulation is where the embryonic disc starts to split into a little stalk here that connects it to the uterine wall, and then a disc. And that disc itself forms 3 layers. So if we just go back to the previous picture, where the difference from before is, previously the blastocyst was encased within, or certainly on the surface of, the uterine wall. Now that it's developed further, it's got larger, there's been more cleavage of cells, it started to come away from, but remain attached via stalk, which will obviously develop into the umbilical cord. However, the amniotic part, the amniotic cavity, has stayed where it was, because that's going to be used to develop the placental tissues. But the embryo itself has moved away. So we've got our stalk, which will be attached to the uterine wall, which will be around about here, and then we've got our disc. And the 3 layers we have are referred to as ectoderm, mesoderm, and endoderm. So it's from these 3 layers that all tissues will develop. So you may well hear me refer later on in the module to something be of mesodermal origin or of ectodermal origin. So you know what that means is that at this point, So this sort of 16-day period when gastrulation occurred, this is the cells from which those tissues have developed. So as a general rule, obviously ecto refers to outside, mesio in the middle, endo inside. As a general rule, the things that are ectoderm will be on the outside of the body. Mesoderm will be sort of the bulk in the middle, and the ectoderm will be sort of lining and things. However, it's not quite as specific as that, but it's a reasonable general rule. There are, as with everything, I'm afraid, exceptions to every case. So the ectoderm, you see we're talking about epidermis, so the outside layers, hair, nails, skin, the lens of your eye, teeth, in fact, particularly tooth enamel. It's the outside layer. It's developed from ectodermal tissues. However, because there's exceptions to the issue, you need to remember that the nervous system develops from ectoderm, even though that's right in the very inside of us. It's just the way these things go. So our brain and our spinal cord and the nerves that arise from it, they develop from ectoderm. Mesoderm is pretty fair to say it's all the bits in the middle. Muscle, bone, blood, connective tissue basically, which we'll talk about shortly. The rest of the tooth, so the inside of the tooth, the dentine. I need to mention that this is an American diagram, hence why it says dentin and not dentine with an E on the end, but either way. Organs, so things like heart, lungs, spleen, that type of stuff. Reproductive system. The bulk of it, the mesoderm. You'll notice that mesoderm is the thickest of the three layers. Because let's just highlight these so it's really clear what we're looking at. Oh, that's not a highlighter, that's pen. Let's go back and find the highlighter, shall we? This might be a little bit large. But... So, yeah. We're looking at the shaded area here, okay? That's your ectoderm. Then the gap between the two in the middle is the mesoderm. And then the area that's in black here is your endoderm. The space either side, let's go with blue, here and here, is the yolk sac part, so the part that's producing energy at this point, because previous to this point there hasn't been circulation, so the cell, all these divisions have been happening from its own energy source. That's the remainder of that, and that will gradually reduce as the ectoderm, mesoderm, and endoderm becomes larger. So the point of doing all that was to say that the mesoderm is the largest of the three layers, and therefore most tissue will come from that. Hence muscle, bone, spleen, the heart, lungs, that type of thing. And then the last section is the endoderm. Generally the endoderm is lining, so linings on the inside. We've got the linings of the lungs, the lymphatic tissue like your tonsils, bladder, your digestive tract, your urinary tract, glands, things like that, so right on the inside. So it's a fair assumption that if it's on the outside, it's come from the ectoderm, and if it's on the inside, it's come from mesoderm, and if it's a lining, that it's endoderm, but it's not an absolute set in stone thing. So that all occurs as part of gastrulation, which is happening at about 16 days post-fertilization. So from then on, things continue to develop. As I said, the first trimester is by far the most involved trimester. It's a time when most things happen, and it is up to week 13 of pregnancy. The trophoblast, so remember the outside part of the blastocyst that attach to the uterine wall, that goes on to develop the extra embryonic structure, so things like the placenta and continuation of the umbilical cord. We've got facial development, which includes the teeth. So the primary teeth at this stage, the baby teeth. That all occurs 4 to 12 weeks, so all in the first trimester. I have put some additional reading on Blackboard for you. Now, you'll talk about this an awful lot more when you do the oral biology module next semester. So it's probably quite advanced at this stage, but it's there on Blackboard for you if you want to have a look at it. It gives you sort of a step-by-step, week-by-week of what's happening in the mouth and the oral cavity from 4 to 12 weeks. And from the 10th week, we stop calling this little being an embryo and we refer to it as a fetus. So from 10 weeks onwards, a fetus. Cells differentiate, so instead of just having a big blob of identical cells like we had at the sort of morula and the blastocyst stage, we've got differentiation. They're going to form their specific tissues. And because so much happens in the first trimester, it's really important that the mother is well looked after. The fetus is hugely susceptible to disruption by illness, so infection, things like that, for the mother, and any nutritional deficiencies. So this is a time when women are advised to take vitamin B12 and folate, and also in the run-up to pregnancy if they're planning it. Because obviously a child developing at this rapid rate is going to require an awful lot of nutrition. So if the mother's not taking that in or taking in extra than she normally would, then the baby might be lacking. It's also at this point that the baby is most susceptible to damage as a result of alcohol and drug use, and also radiation, so x-rays and things. Hence why women are told to avoid all alcohol and drugs, obviously, during pregnancy, and where possible to avoid radiation. If sometimes it's essential, obviously, but in general we'll avoid x-rays and things as much as possible. So a huge amount going on in the first trimester. Progressing on to the second trimester, so from week 14 to week 26, we start to see things like skin and hair develop. We can see, this is the point where most women will have their first scan, and generally you might be able to see what sex the baby is at sort of about the 20-week mark. Also at the 20-week mark, the fetal heartbeat can be detected. The fetal heartbeat starts much earlier in the first trimester, however it can be difficult to detect at that point. So second trimester is generally when it's found in a normal sort of ultrasound. There's a lot more movement of the fetus. In the second trimester, the mother will feel the baby move, kicking, that type of thing, because muscle is developing. Up to that point, there may have been limbs, but they couldn't particularly move independently because they didn't have the muscle, but now they do. Respiratory system develops, so the lungs grow and develop, and by the end of the second trimester, the baby is at a point where its lungs could be functioning if required. Very poorly babies can be born at 25, 26 weeks, and they are considered viable. So before they've even necessarily got to the third trimester. And that's because their lungs are at the point where gaseous exchange is possible. So it's possible for them to take air into the lungs and for the oxygen from the air to go into their bloodstream, and from the carbon dioxide from their bloodstream to go back out through the lungs. They may not have the muscle to be able to physically breathe themselves, so they're likely to need assistance, but their lungs can do the job with assistance. Eyelids, as you can see from the image, eyelids are there, and the baby can open and close its eyes, and we get an awful lot of fat start to be deposited. babies need lots of fat. They've got an awful lot of growing to do once they're out into the world, and obviously they have a fat-rich diet initially, but they need a lot of their own fat to use as well. Then in the final trimester, so from 29 to approximately 40 weeks, although they do technically say that the full term varies, and some places will say 38 weeks and some places will say 42 weeks. That's why you might find when you're reading some literature that it refers to week 1 and 2 of pregnancy as pre-fertilization, hence the 40-week pregnancy. But either way, 38 to 40 weeks is considered to be full term. What happens here is basically the baby grows. They lots more fat, they get a lot bigger. All the tissues are there at this point, but they're just getting bigger. The lungs become much more viable. So the baby actually makes breathing-like movements. It won't breathe until it's born, obviously, because it's in a fluid environment. It also doesn't need to breathe until it's born because it's got getting the oxygen and nutrition from the umbilical cord. But they start to make movements as if they're almost practicing breathing. We get sensory connections in the brain forming. So babies who are born very preterm, so that early, very early third trimester stage, often have issues with their senses. So they might have sight or hearing issues. And that's because the sensory brain connections occur in that third trimester. The fingernails come to the end of the fingertips. So babies that are born preterm might not have fully formed fingernails, but by full term, the fingernails are the end. And like I said, 38 to 40 weeks considered full term, baby's ready to be born. So that's very much a whistle-stop tour through pregnancy, and any of you who've got your children of your own will probably know an awful lot more about that than what we've talked about there. But it's just to give you a rough idea of what's going on, where the tissues have developed from that we're going to go on later to look at and potentially treat if disease should be occurring. So now I want to have a think about different types of tissue. We're looking at now at a fully formed human, okay, so not in utero anymore, a child that's been born, child, adult, whatever. All humans are made up of 4 types of tissue. Those are muscle, nervous tissue, epithelium, and connective tissue. So muscle, I'm not even really going to mention now. All these types of tissue we're going to talk about a lot more in the individual lectures, but muscle in particular is really, really complicated. You know, there's different types of muscle. You've got smooth muscle, you've got skeletal muscle, and you've got cardiac muscle, and they all work very differently and do very different jobs. But it is an individual tissue type on its own. Now, what we're looking at here is nervous tissue. Let me just get a cursor. So nervous tissue is the tissue in your brain and the rest of your central nervous system, so the spinal cord. So you can see at the top of this image here, we've got a brain. Zoom in a little bit. And then we've got the spinal cord that runs all the way down the length of the torso. So that's your central nervous system. And then branching off from there, we have the peripheral nervous system. So the nerves of the neck, of the thorax, down each arm, of the abdomen, pelvis, and then down the legs. So it's a very specific type of tissue. It's a tissue that's able to both generate and conduct electrical impulses. And that's how the nerves pass from our brain, which is the ultimate control panel, to the rest of our body. Again, we've got a whole lecture on nervous tissue, and we can talk about that in more detail. And then we've got the other two. We've got epithelium and we've got connective tissue. Epithelium is a lining tissue. Now that's both external and internal. So the epithelium externally is your skin. But the epithelium internally is what lines all the cavities. So it starts in your mouth and it goes all the way down and out the other end. There's also epithelial tissue in your glands. Then, connective tissue is everything else in between. So if it's not muscle, and it's not nervous tissue, and it's not epithelium, then it's connective tissue. So I've got two diagrams here, well, two histological slides actually from a microscope of the interface between epithelium and connective tissue. It's looking at a specific type of epithelium, a stratified epithelium, which means it's got multiple layers. But either way, the top diagram, so the top layer, okay, all this stuff here, up to this squiggly dark line, okay, that's your epithelium. Underneath the squiggly dark line is what's referred to as the dermis here, or connective tissue. And you can see the sort of the connective tissue has sort of little finger-like projections into the epidermal layer to anchor them together. And then similarly in the second diagram, I'm not going to highlight this one because it's probably a bit more obvious, but you can see you've got all the epidermis in the darker stain, and then connective tissue in the lighter stain. The thing that's worth noting here is that in the darker stain, can you see there's an awful lot more cells? What stains is the nucleus of the cell. So in here you've got loads of nuclei really close together, whereas in the connective tissue they're a lot more sparse. And that's one of the main differences between epithelium and connective tissue. The epithelium has got close, tightly packed cells. There's very little intracellular space or any kind of matrix between them. So they're really, really tightly packed together. And they all lie on what's called the basement membrane. So see this dark line that we have on the top here? That's your basement membrane, where they're incredibly tightly packed together. Underneath the basement membrane, we've got the connective tissue. Now, the epithelium does not have a particularly good blood supply, in fact, at all. It relies on nourishment coming from the connective tissue underneath, because that's well supplied. Hence why, when you've got layers of epithelium like this, you'll notice the cells at the bottom are much bigger and fuller looking, because they're getting all the nourishment, whereas the cells at the top are thin and they're actually heading towards the surface to be shed off and die. Or the other way around even, die and shed off. Connective tissue is the most abundant tissue in the body. The cells, as you can see from this slide, are much wider spread apart, and there's a lot of intracellular stuff in there. So fluid, matrix, it's called mucopolysaccharide ground substance. Again, we'll talk about that more when we talk about connective tissue. That basically fluid and a sticky fluid type material that suspends the cells within it and contains all the nourishment that the cells need. It's sort of, most often is jelly-like, but it can become more rigid. So, for example, cartilage and bone are actually a type of specialised connective tissue, and they're obviously not jelly-like. So just to give you a very rough idea of the different types of epithelium and connective tissue, epithelium is referred to in two different categories as either being simple or stratified. So simple epithelium is just one cell layer thick. You've got connective tissue, you've got a basement membrane, and you've got one cell of epithelium. And you generally find that in places where you need things to be absorbed. So, for example, in your digestive tract, you're looking for materials that are in the hollow of the tube to absorb out into the tissues. So you don't want a long way for them to go. So that's simple epithelium, and different shapes of cell will give it a different name, but we won't go into that just yet. The other category of epithelium is called stratified, and that is multiple layers of epithelium, and that's what we were looking at in these slides here. Multiple layers. They start off as big, fat, healthy cells, and then they work their way towards the surface to become thin and shed. And stratified epithelium is found, oh, sorry, wrong way. Stratified epithelium is found in areas where you need more protection, areas where you might have friction or exposed to drying. So, for example, your skin is stratified epithelium, otherwise you would lose all the moisture from your body. Similarly, in your mouth, you've got stratified epithelium, and that's because when you're eating and drinking, you need protection. So they're the two categories of epithelium that we'll talk more about in your next session. Connective tissue, loads of different options here, and I don't really want to confuse things, but suffice to say it's everything else, like I said before. It's adipose tissue, which is fat, fibrous tissue, so strong, sort of leathery type stuff. Elastic, so stretchy. Your blood is a specialised type of connective tissue for transport, as is lymph, bone, cartilage, all this kind of stuff. So there's a lot more to connective tissue, and I don't think we need to talk about it. At this point, we've got a lecture coming up about that. So that really brings us to the end of what we're going to talk about in this session. Just going to come back to the learning outcomes so that we can just be clear in our minds that we've covered everything. There were just the three, but they're obviously quite wide. So give a basic description of embryonic development, explain development of ectoderm, mesoderm and endoderm, and give a basic description of the different types of tissue. Please be clear on the use of the term basic here. By all means, go ahead and do some extra reading, but the volume of information available is vast, and I don't want you to get overly complicated and bogged down. This is one lecture of the entirety of this module, so try not overcomplicate things. Basic. If there's something that's particularly interested you or something that you've struggled with in this session, then by all means do a wee bit of extra reading on that to clarify it for you. But try not and get too in depth, because you could. This is a whole subject in itself, a whole degree or master's or PhD of its own. I've put some references there for you. Obviously, there's the Ross and Wilson textbook. The other two, the Avery and Berkovitz books, are not available as ebooks, unfortunately, but there are books available on each site, and they are quite useful just to have a read of the section about embryology and tissue types. Avery was particularly good for tissue types. So if you want to borrow those books from each site, then you could either go to the UHI library in Inverness College or Lewis Castle College, or if you can't get them there, then if you approach one of the tutors in your site, they may have a copy. In fact, they will have a copy that you can look at. You won't be able to take it away because they're our own personal copies, but they are there. As we already know, the Ross and Wilson is available online as an ebook. So that's us done. I'll see you in class for our tutorial where we can go through this, test our knowledge a little bit and discuss any issues that you've had. Thank you very much. Bye-bye.